everyone, and welcome to our final video in the protein trafficking series. This video will discuss transport by vesicles of proteins to their final destinations and the role of signal sequences. When proteins have reached their correct location within the TGN, the membrane at those locations buds off into vesicles. More than one protein can be contained within each transport vesicle. The targeting of a protein to its final destination is controlled by its signal sequence or its lack of signal sequence. A signal sequence is a short stretch of amino acids that indicate where in the cell a polypeptide belongs. Signal sequences work in a very particular way. A signal sequence will bind to a specific receptor protein at the surface of the organelle, and a channel will form in the organelle membrane. This channel allows for the targeted protein to move through the organelle membrane into the organelle. There are some proteins that do not contain a signal sequence. If a protein does not contain a signal sequence, it will remain where it was synthesized, in the cytosol. Each signal sequence is quite different from the others and quite incompatible to ensure consistent targeting to the correct compartment. There are many possible final destinations for these vesicles, as determined by each signal sequence. Some of the most important destinations are 1. The plasma membrane. A vesicle containing proteins travels to the plasma membrane. There, the vesicle membrane opens up and fuses with the cell membrane. So, all the proteins embedded in the vesicle membrane become part of the plasma membrane, and the proteins being carried in the vesicle are secreted from the cell itself. 2. A lysosome. A vesicle is transported to a lysosome, and the lysosome fuses with the vesicle membrane. Digestive enzymes in the lysosome essentially eat and digest the proteins in the vesicle. This process is called autophagy, or cell eating. 3. A mitochondria. A vesicle is transported to a mitochondria, and the vesicle membrane opens up towards the mitochondria and fuses with the mitochondrial membrane. Therefore, the proteins in the vesicle become part of the mitochondrial matrix, and the proteins in vesicle membrane become part of the mitochondrial membrane. There are many other destinations, including the ER, the Golgi, and many other organelles, but these destinations are less important.